Welcome. We are so happy to have you for day 5 of the ASF Virtualpalooza. You are in store for a great one tonight. Thank you for joining me on a Friday night. I thought since we're doing this for the next 2 weeks, I would start wearing in the next 2 weeks for every Virtualpalooza some of the awesome t-shirts that you guys have created. You are the most innovative and creative community when it comes to t-shirts, and I can't stop buying them, even though my husband's like, please, Amanda, stop. So tonight I'm supporting the Cami Can t-shirt. So I don't know if you're on, but Cami, I'm supporting you tonight, and stay tuned for Monday to see the next t-shirt I'll be supporting. So like I said, you're in for a really great treat tonight. If you're joining us for the first time for the ASF Virtualpalooza, we're just so happy to have you. A couple housekeeping, a couple reminders before we jump right in is, one, there is a question poll, a question area on the screen throughout the presentation. Please feel free to ask questions. We are going to do our best to get to as many questions as we can tonight, because this is going to be an amazing presentation. I'm sure you're going to have questions. Whatever questions we can't get to, we will make sure that we will answer those for you and get them out in a PDF after the presentation. And just so you guys know, the way that we're going to work this is every week we will upload all the videos and the PDFs and the presentations that we have up into the ASF's website, so you'll be able to keep track and rewatch them. So stay tuned for that. Also, because you've come tonight, you are going to get another awesome t-shirt to add to your collection. As a thank you from us, you will be getting an ASF Virtualpalooza t-shirt mailed to your home, just to thank you for being a part of this, from our awesome sponsors at Ionis Pharmaceuticals. So we're so excited to be able to do that for you. Also, I can't not say, please make sure, if you haven't yet, sign up for the ASF National Walk. We are so excited this year. It's going to be virtual. Although we'd love to be in person, we need to be safe, but we've got an awesome virtual event planned for you. So if you haven't signed up, it's not too late, and it's not too late to get your t-shirt. So please make sure you sign up. So I'm sure none of you want to hear me keep talking, because you came here tonight to hear a rock star. We're so excited to have Dr. Becky Burdine with us tonight to talk everything genetics. I know that this was something that I needed, especially early on, and even 3 years into it, it's still very confusing to me. So it's always good to have these great conversations and these great talks. Not only is Dr. Becky Burdine just amazing at being able to articulate this, she's also a mom to Sophie, who is 14 years old, who's just an amazing individual. She's also part of the ASF Board of Directors and the ASF Scientific Advisory Board. She's been a part of this community and a part of this work for such a long time, and we're so lucky to have her. So I'm going to go ahead and let's shift over to presenter mode to Becky. And let's give a round of applause and welcome Dr. Becky Burdine tonight. Becky, if you want to go ahead and share your screen whenever you're ready. There you go. Welcome.
Hi. So after that introduction, I'm feeling a little bit stressed. So I'm going to try to do my best here. I wanted to thank all of you for coming, and thank Amanda for giving me this opportunity to talk to you. I wanted to tell you a little bit about me before we get started. As Amanda mentioned, I am the mother to this little girl here, Sophie, and she's just been the joy of our lives for 14 years. And some of you may know me and her, but some of you are probably new to the community, and I'm hoping that some of you are, because part of what we're trying to do tonight is make sure that you understand what the genetics are behind Angelman syndrome, and how that helps us understand some of the therapeutic approaches that are being taken right now in our community. So in addition to the disclosures that Amanda mentioned with the ASF, I also need to let you know that I am part of the Ovid Clinical Trial Steering Committee. And what this means is I'm part of a committee that includes other physicians and other scientists. And our job is to oversee the trials that are going on, STARS, ALARA, Neptune, look at any adverse events that happen, provide basically oversight back to Ovid about the clinical trial, and help look at the data when these trials are over with. So I've been working with them now for quite a few years, and in truth, since some of you may be beginners, anyone who speaks to you in our community that has these kind of interactions or these sort of relationships should really tell you about them, so that you know any information you're getting, there's also those things behind them. All right, so what are we going to do tonight? Well, it's Genetics 101. So what we're going to do is first, I'm going to give you a general overview of the genetics behind Angelman syndrome and the different ways that Angelman syndrome is caused. Then we'll have a very brief discussion about something called genotype phenotype. What that means is the way that the genetics causes Angelman syndrome, and then how that manifests in the way that your child presents with Angelman syndrome. So genotype is the genetics, phenotype is how they present. I will then give you my impression on where you should focus as a beginner. So if you're new to this community, you're new to Angelman syndrome, what are the things that you should probably focus on? Because I'm sure, like all of us when we first started in Angelman syndrome, the information overload is incredible. There's so much out there. There's so much out there that sometimes gets conflicting. Lots of people want to give you advice. So what do you do? Where should you start? And I'll tell you my thoughts on that after having been doing this for 14 years. You can take it as you like. I'll then stop and ask if you have any questions, and we'll do a little question and answer period then. And then if we have time, what I'll do is move on and explain to you why the genetics tell us that we can develop better therapeutics. And then finally, what I think you might need to know about clinical trials in a general sense. I'm not talking about the various different companies and what they're doing. There is a phenomenal video already on the ASF website that came from all the industry presenters on Monday that will tell you what they're doing and how they're doing it. I suggest you go look at that for those details. I just want to tell you about clinical trials and a little bit about the experience that I've had learning about them being part of OVID's Clinical Trial Steering Committee. All right. So first, to understand the genetics of Angelman syndrome, we have to do a little bit about genetics. And the first is to understand that we have 23 pairs of chromosomes in our cells. So every cell in your body has 23 pairs of chromosomes made of DNA. And this is a karyotype. This is actually my son's karyotype. These are all the chromosomes. And the one that we're going to be talking about the most tonight is chromosome 15. And with the chromosomes, you get one from your dad and one from your mom. That's how we get our 23 pairs. 23 chromosomes from mom, 23 chromosomes from dad come together to make us who we are. And the chromosomes are made of DNA, and that DNA is set up into units of information. And those units of information are called genes. So when we talk about genes, we're talking about units of information in DNA. And in the case that we're talking about tonight, what we care about is chromosome 15, and we care about this area in red. The DNA in this area is encoding a gene called UBE3A. And that information can be used to make the protein, also called UBE3A. All right, so what are we talking about with genes and proteins? This is the central dogma in molecular biology. So you may not have thought when you logged on tonight that you were going to become molecular biologists, but we're now indoctrinating you into this. So as I said, with the chromosomes are made up of DNA, and DNA has information encoded in it, just like a book. All of the information encoded in DNA is made up of 4 different letters, essentially, A, C, G, and T. And those 4 letters are used in different combinations to encode DNA information that your cells can use. One of the ways they use that is they have proteins that read the DNA and they make message RNA out of it. So this is this information comes directly from the DNA. It has the same information. And then there are additional proteins that can read the RNA and read the information and use that to make a protein. So RNA is read in series of 3 letters. You don't have to worry about the details of this necessarily, but what will happen is the protein that reads the RNA will see, say, for example, this code, this word, and it says, I know what that word is. I'm going to put this amino acid in the protein. And then it goes to the next word and says, well, here's this word. I'm going to put this amino acid in the protein. And it does this on and on and on until it puts together a protein made of amino acids. This is the central dogma in molecular biology. DNA goes to RNA. RNA is read to make proteins, and proteins are the machines that make your cells work. You can think of proteins in general as machines. They're machines. They read the RNA like a machine. They read the DNA like a machine. They work together to do various different tasks in the cell. They're also signals. So for example, when you get sick, proteins are used to signal different parts of your immune system to tell it to do its job. They're also structures. So your hair, for example, is made of a protein called keratin. So we use keratin to build the structures that make up things like your hair and your nails. So all of our cells are basically little compact units filled with this information that gets read, gets turned into proteins. The proteins go on and do their jobs in the cells, and that's how all the cells do their functions. So the cells in your gut are containing the kinds of proteins they need to take up nutrients and to break them down so you can use them to make more protein. The cells in your brain, the neurons, are using different sets of proteins to receive information and send that information on so you can do things like listen to this webinar, or I can do things like move my hands around when we talk. So when we talk about our gene of interest here, which is UBE3A, the chromosome has the DNA with the information to make UBE3A. The DNA can be used to make an RNA that has the information to make the protein, and the proteins can use this RNA to make protein that is UBE3A itself. That's the general idea with the molecular biology at this point. Now, importantly for Angelman syndrome, you have to understand something that's really very cool if you're a scientist. If you're a geeked out scientist like me, this is the coolest thing ever. So we receive one set of chromosomes from mom and one set from dad. And just for ease sake, mom is going to be red in this presentation, dad's going to be blue. And what happens is mom has eggs that have one set of her chromosomes, so the 23 chromosomes. Dad makes sperm that has 23 chromosomes. And during fertilization, these two come together to make the zygote, which is going to turn into the baby, which has now 23 pairs of chromosomes. The thing that's really cool is that your cells know which chromosome came from mom and which chromosome came from dad, and they know that for the entirety of your life. So right now in your body, your cells know which of your chromosomes were your mom's and which of your chromosomes were your dad's. And we don't have to go into the details of how cells know that, but it is really interesting how they do it. And it's not something we have 100% understanding of yet. And for the most part, it doesn't make that big of a difference, because as long as you have one good gene, either on mom's side or dad's side, you're fine. Most genes, as long as you have one functional copy, you're fine. Angelman syndrome is different, and that's why we need to know this one piece of information. So in a typical situation, we're going to talk about a neuron specifically in the brain. Here's our chromosome 15s. You've got one from mom and one from dad, right? Because you have the pair. UBE3A is encoded in this region. But neurons know which chromosome was mom's and which chromosome was dad's. And so the chromosome that was mom's is set up in such a way that it can make the UBE3A message, and that can be made and encoded into UBE3A protein. Dad's is perceived differently by neurons. When neurons see dad's chromosome, they make something different off this chromosome, and that prevents UBE3A from being functional. So in a typical neuron, we're all making UBE3A from the chromosome we got from our mother. We're not making any to none, little to none from the chromosome we got from our father. This is called imprinting, and that means when a gene's expression and ability to be turned into a protein are controlled by whether or not the chromosome is maternal from mom or paternal from dad. In Angelman syndrome, your chromosomes throughout the vast majority of your body, everything but neurons, are not read this way. Most cells don't care if it's maternal or paternal. They express UBEA through both chromosomes. But when your neurons become neurons, when they become specialized to be neurons, they now listen to this information and they turn off dad's UBE3A and they only use mom's. So this is critical, because what it means is in the brain, we only have one good copy of UBE3A. And we know UBE3A is very important for the brain to function. And so what happens in Angelman syndrome is we lose the ability for this to be expressed off of mom's chromosome. So before I talk about the different ways that we can lose UBE3A, I want to talk about what UBE3A actually does. There's been a lot of work into what the function of UBE3A is in cells. I'm going to tell you about the main function that we know of with UBE3A, and that is this one. So in this diagram, these are all proteins. So these are all little machines that are set up to do their jobs. And this green one here is the UBE3A protein. And its job is to take another protein called ubiquitin and it's called ubiquitin because it is ubiquitously expressed, and that's where it got its name. Ubiquitin is recognized by UBE3A, and it is placed onto other proteins that UBE3A recognizes. As it continues to place ubiquitin onto these proteins, that tells the cell that this protein needs to be recycled. It's time for it to go into the trash and be gone. So the role of UBE3A is to basically signal when various different proteins in the cell need to be degraded and gotten rid of. And the way that happens is once these proteins have that ubiquitin signal on it that they get from UBE3A, they're sent to something called the proteasome, which essentially tears them up into their individual amino acids so they can be used for making new proteins. This process is essential for neurons to be able to function optimally. And we know that because in Angelman syndrome, when we don't have UBE3A functional, neurons don't function optimally. And we know that from looking at mouse models. We know that from looking at neurons in dishes. And so one thought is the reason that neurons may not function optimally is because they have too much protein in there that needs to be degraded. So there's a lot of garbage basically sitting around and it's mucking up the works, if you like. That's one thought. There are some other thoughts about what UBE3A might be doing, but this is its major function. And so in all of your cells, UBE3A is doing this job. In neurons, this seems to be very essential for neurons to be able to work. So we really want this protein and we want it to be present in our neurons. All right, so how do we lose it in the neurons? Well, there are a number of different ways that we develop Angelman syndrome. And so I'm going to walk you through those so that you understand, and hopefully you'll be able to recognize what your loved one has for their particular genotype. That means the genetic changes that cause Angelman syndrome. So again, with my picture, we've got the female chromosome here, mom's chromosome and dad's chromosome. Remember, on moms, we can make the protein, on dad's, we do not. And we'll talk more about why that is in a little bit. If you hear a lot of noise behind me, it's my cat who is completely convinced he must be present in this webinar. I'm going to try to ignore him. Sorry about that. All right, so the most common way that we form Angelman syndrome is through a deletion. So mom's chromosome is missing that region that normally has the DNA that encodes for UBE3A. And so in this case, when the neurons form, they don't have UBE3A on mom's chromosome. They don't recognize it as being present on dad's chromosome. And what we end up with is a situation where we don't make any UBE3A in neurons. Excuse me one second. Okay. This is the most common form of Angelman syndrome, and it's due to a deletion. So now what I'm showing you are what the deletions look like. I've turned the chromosome on its side. So this is the same chromosome from here, but now I've turned it sideways to show you what we've done here. And what we've done is we've taken this region that's usually deleted in Angelman syndrome and blown it up so that you can see what it looks like when you look at the different genes that are present in this region. So the BPs stand for breakpoints. These are the places where we know there are often breaks in the DNA that cause deletions. And you can see that there are numerous ones shown here. BP 1, 2, 3, 4, and 5. These are all regions that are known to have been broken and deleted in Angelman syndrome. Anything that you see in green, so these are different genes like NIPA1 and 2. Down here are the GABA receptors. These are all different genes that are expressed both from mom's chromosome and dad's chromosome. So typically, as long as you have one chromosome that can express these genes, you're fine. The things that are in blue are specifically expressed from dad's chromosome, but not mom's. And the two genes in red are expressed from mom's chromosome, but not dad's. And you can see that includes UBE3A. So when we have deletions, the two most common types are class 1 and class 2. We're removing these regions, all these genes. Most importantly, though, we're removing UBE3A, and that's what causes Angelman syndrome in the deletion cases. The class 2 deletions are a little bit smaller. They remove fewer genes. The class 1s are a little bit bigger. I will just say, although I'm not going into the details, we have class 3 through 4, 5. They're all different sizes. So you can have deletions that come from any of these breakpoints. As long as they remove UBE3A, when you delete that DNA, it will cause Angelman syndrome when it's inherited from mom. I also want to point out that these are microdeletions. What that means is this is a really small deletion. And that might sound really funny to say, because for us, this sort of looms large in our lives. We've deleted this piece of DNA and now we have Angelman syndrome. But in truth, it's a very tiny piece of DNA. The class 1 deletions are about 6 megabases. And that sounds huge. Megabases, that's really huge. But keep in mind the human genome is 500 times that. And we have about 20,000 genes. And really in this deletion, the only one that matters for Angelman syndrome is UBE3A. So keep in mind that you still have 19,999 other genes that are in there making up who your child is. We focus a lot on the loss of this one, of course, because it causes Angelman syndrome. But I think it's good to sort of keep in mind how tiny this is and how many other things are in there that are important for your child and who they are. All right, those are deletions. And like I said, that's 60 to 75% of the cases. The next class I want to talk about are mutations. So with a mutation, what happens is that information that's on mom's chromosome has been altered at the DNA level. And because of that alteration, the protein that can be produced from that information doesn't work. It's non-functional. Now, at this point, I'm going to get into a little bit of some technical stuff about how mutations work. This is if you're really interested. If you're not really interested and all you want to take away from this is mutations cause there to be a UBE3A protein that's not functional, that's all you need to know about why these cause Angelman syndrome. If you want to know a little bit more about this, like you've looked at your genetics reports and you see some words that you would like to understand, see if you can follow through with this one. All right, so what we're going to do to explain mutations is this. I told you before that the DNA encodes through these letter codes, these triplets made up of these 4 letters, the instructions to make protein. So these are going to be our examples. This is a typical example. You can think about the DNA as a book and the code as words that are in the book, and what the words say are the information that would be a protein. So in this case, our analogy is going to be this book for DNA, and this is the information, and that information says the UBE3A gene encodes a protein called a ubiquitin ligase. So what I told you before was that UBE3A can add ubiquitin onto proteins. So we use the word ligating for adding in proteins. So it's adding ubiquitin to other proteins. That makes it a ubiquitin ligase. All right, now what happens a lot of times in DNA are there are changes that don't really mean anything. And what I mean by that is if I took everyone that's on this webinar right now and sequenced all of our UBE3As, they would all look really different from one another. Even though they're all functional, they would all look very different. And that's because DNA is prone to picking up changes, and most of the time those changes don't have much of an impact. When we see a change that differs from what we normally see in a population, we call that a polymorphism. And as I said, it doesn't impact the protein, but here's one of the ways this could work. So there's a change in the DNA in this particular case, and it has changed this piece of information from ACG to ACC. But the thing is, the cell reads those as being exactly the same thing. So you still make the exact same protein. That's called a polymorphism. It's a change in the DNA, but it doesn't impact the protein. And so in our example, we could do this. We could say, well, the UBE3A gene encodes a protein known as a ubiquitin ligase. In terms of the information in our first sentence and our second sentence, they're the same. It's a little bit different, but the information overall is the same. And in this case, you would still get a functional UBE3A protein. So if you see polymorphism in a genetics report, it just means there's a change, and most of the time those don't have any consequence. What does have a consequence are changes in that DNA that impact how the protein gets made. So in this case, this is considered a missense mutation because now we're going to change the meaning of some of that DNA. And in this case, we've changed this CUU to a CCU, and the cell reads that differently. And it would put a different amino acid into this protein. And the problem with that is that, as I said, proteins are like machines, and machines work because they are in the right conformation to do their jobs. When you change the sequence of proteins, many times what happens is the protein ceases to be able to do its job. It can't recognize other proteins, it can't carry out its needed jobs in the cell, it becomes a non-functional thing. And in this case, that's what would happen. So in our example, what I've done here is I've still got the sentence, but now it doesn't make any sense compared to what it should say. It should say it encodes the protein. Here I'm saying it explodes the protein. Totally different meaning. This is not a useful sentence because this is not a useful protein. That's a mutation and a missense mutation. Many in our community, though, have something called a nonsense mutation or a stop codon. What happens here is the DNA changes, but it makes a signal that proteins recognize as a signal to stop making protein. So it's reading the RNA, it's making the protein, it gets to this signal and says, oh, I'm done, and it stops. So you don't make a full protein. You don't make one that can do its job because most of the protein isn't there. And so in this case, it would be like truncating our sentence. The UBE3 gene encodes a protein called period. So we've lost the information because we didn't actually finish the sentence. It's very analogous to this where we didn't finish making that protein. I just want to mention here that there are mutations that either put additional sequence in or take sequence away. Don't confuse deletions that are within the coding region as the big deletions we talked about before in class 1 and class 2. If you have a very small deletion that takes out, say, 2 or 3 of these nucleotides, you will change the information so that it can no longer make the protein. If you add a nucleotide in, it will also change the information. And that's because, as I said before, proteins read RNA in groups of 3. So if you add in letters or take letters away, the groups of 3 change. And when the groups of 3 change, you start to make proteins that don't look anything like what they should because they're reading the sequence completely wrong. All right, so it is a little hard for me because normally when I give these talks, I can see everyone in the audience either nodding or shaking their head, so I know I've completely lost you or not. So I'm going to assume I haven't lost you and we'll keep going, but we're going to get to the Q&A part, and if you want to come back to any of this, I will do so.
Hey, Becky.
Yes.
Do you mind if I just, there is a couple questions I think might be good to answer before we move on, if that's okay?
Sure.
Yeah, so just one of the questions someone asked about, does the size of the deletion matter? I don't know if you can speak to that.
I'll talk a little bit about this in a bit.
Okay.
Yes and no. In general, smaller deletions tend to have less of an effect than bigger deletions.
Okay.
That is not a truism, though. There are plenty of children who have, and loved ones, people with Angelman syndrome who have smaller deletions that are as strongly affected as people that have bigger than class 1 deletions. So it's not a given that the size of your deletion will dictate what you can and can't do as an individual with Angelman syndrome.
And can you give any advice to people on, a lot of individuals have the diagnosis of Angelman syndrome but may not know the size of the deletion?
Okay. So if you've been diagnosed in the last, probably last 8, maybe 10 years, it probably will have some information about the size of the deletion. It might be hard to tease out of your record,
Sure.
but it will be there. If you were diagnosed, let's say before 2005, it probably won't. And that's just because at that time, the genetic testing that we have didn't get this detailed. They would just look and see if I go back to here, what they would do is they would take blood or they would do a sample. They would look at the chromosomes spread out like this, and they would look to see if a probe for UBE3A would light up both 15s. If it wouldn't light up one of the 15s, but it lit up the other, then they would say you have a deletion. But it would take additional testing to figure out how big that deletion is.
Do you think?
Recently,
oh, sorry. Go ahead. Sorry.
Sorry.
No, go ahead.
More recently, the tests will tell you how big that is. They'll tell you what the breakpoints are. If they tell you, if they say it's between breakpoints 1 and 3, for example, that's a class 1. You can also go by the size. If they're 6 megabases or a little bit bigger, it's class 1. If they're 5 megabases or less, they're probably class 2.
Do you, from just your experience of what you know, and then we'll let you get back to it, is it important for people to understand their size of deletion or to know it as we're moving into clinical trials?
In my opinion, no.
Okay.
So the only reason it's important to know what your genetic makeup is for Angelman syndrome is because we need the proof that your child does have Angelman syndrome. And like I said, now it's not such a problem, but when Sophie was first diagnosed, there were people who thought they had Angelman syndrome, but they didn't have any way to know. And in some of those cases, it turned out that they didn't have Angelman syndrome. They had GRIN3B or they had Pitt-Hopkins. So we need the molecular diagnosis to know for sure that we're doing Angelman syndrome, because you can imagine if you don't have Angelman syndrome, some of these therapies would be very bad to have.
Absolutely.
So you need to know that. Whether it's a class 1 or a class 2, I don't think it's that useful. I think as parents, of course, we want some reassurance. And I think if you know your class 2 deletion, you might feel a little bit less worried about seizures, or you might feel a little bit less worried about some things. But like I said, it's not a given. There are plenty of children with class 2 deletions that have seizures that are identical to those in class 1.
Absolutely.
So I think if you have a deletion, it's a deletion.
Thank you for answering those. I appreciate it, Becky.
Sure. Was there another question or?
No, you can go ahead. Go on. Yeah.
All right. Okay, so we've got our deletion and our mutation. The next two have to do with the fact that the cells know the difference between mom's chromosome and dad's chromosome. So there are two of these, and we're going to do them together. One of them is called uniparental disomy, or UPD. The other is an imprinting defect. So in UPD, this is actually a very miraculous thing. So when the fertilized egg first comes together, that cell, because it's a cell, looks at all of its chromosomes, and if it sees that it's missing one, in this case, it's only got one chromosome 15 from dad, somehow mom's didn't make it into the egg, in order to survive, it will duplicate dad's chromosome so that you can have a living child. That's really amazing if you think about it. The cell looks it over and says, this won't work, I need 2 chromosome 15s, I'm gonna make 2. It just happens to make 2 out of dad's because that's what it has. That's fine. And we have amazing children out there that are UPD. The problem, though, is that the neurons recognize both of these as being dad's, and they don't turn UBE3 on either of them. So you have Angelman syndrome. Imprinting defects are similar, except that with an imprinting defect, what's happening is that even though this is mom's chromosome and UBE3A is there, the cells don't correctly read the code that's on the chromosomes, and they don't recognize mom's chromosome as mom's. And so they don't turn on UBE3A like they should. In both of these cases, either one, because mom's isn't there, or because mom's isn't recognized correctly, you have Angelman syndrome because you don't make UBE3A protein. Okay, so I've been talking about neurons and non-neurons and mom and dad's chromosomes and which is which. So as I mentioned, in cells that are not neurons, in a non-neuronal cell, so in your skin, your gut, your blood, your eyes, what's happening is that you have mom's chromosome, maternal, and dad's paternal, and they're both making UBE3A. Now, when you see a diagram like this, what this is it's showing you the 2 chromosomes, and these little boxes, this is molecular biology code for a gene. There's a gene here that happens to be UBE3A. There are some other genes here that are not UBE3A. It's not labeled right here, but in this case, we have the genes on moms and dads. This arrow that's going up and pointing, what that's saying is that there is information here that you read from right to left in this particular case, and that information is Angelman syndrome's UBE3A. And you can see on both mom and dad's. This arrow is saying there's some information from this gene that gets read in this direction. So you can see that they're already going at each other in opposite directions. All right, but in neurons, again, it doesn't matter because we express both of these genes. So what happens in the neurons that makes this such a problem? Well, what happens is this arrow gets really strong. So these genes on dad's chromosome get expressed very strongly, and they basically run into the machinery here, and they prevent dad's UBE3A from being expressed. Still expressed from mom's, but dad's is making this big long thing. You can think of this as essentially like a train. This is going very strongly. It's like a freight train, and it's running into the gene down here and preventing it from making RNA. That's really an oversimplification, but it's good enough for what we need. Because dad's chromosome is making information going the other way, it can't make UBE3A. So we have dad's going this way and mom's going this way, and that's good enough for us to get what we need in neurons, unless we have this deletion or UPD or a mutation. All right, so for example, in Angelman syndrome now, with a deletion, we're missing all of those genes from mom, and dad's can't express because it's making this big long transcript. This transcript is referred to as a lot of different things. Sometimes it's referred to as the antisense transcript, or the SNRPN transcript, or the SNRF transcript. In any of these cases, it is a long RNA transcript that gets made, and because it's getting made, you can't make UBE3A on that same chromosome. And I wanted to point this out because this genetic imprinting, this is back in 1989. It was published by Mark Lalonde's group. And this was the first indication that the fact that cells can tell the difference between mom and dad's chromosomes was going to have something important to do with health. And that's because they realized that individuals that had 2 chromosomes from mom, so maternal uniparental disomy, had Prader-Willi syndrome. But if they had 2 parental chromosomes from dad, they had Angelman syndrome. And the region that was important was the same region. So I just wanted to go back and show you this. So in our case with Angelman syndrome, we're making this gene, we're not making UBE3A. In Prader-Willi, it's the opposite. So what happens is that we're making mom's UBE3A, we don't make this long transcript, and that causes Prader-Willi syndrome. So these are considered sister syndromes because they're caused by the same region of DNA, but for different reasons. So that's more of a gee whiz for you to share with friends. I think we've also realized in the community that Prader-Willi syndrome, if you've never heard of it, it's typically thought of as the syndrome where the children cannot resist the ability to eat. If there's food present, they will eat it. They get very anxious about the fact that they have food that they can't access. So there's stories about locking up the food, being very careful with schedules. Well, we now realize that there are some children with Angelman syndrome that have a very similar sort of anxiety around food. And then there are other kids with Angelman syndrome who refuse to eat at all. So we have both ends of the spectrum. But the similarities with Prader-Willi haven't really been recognized until more recently. We still don't understand why these similarities exist. We just know that this region is incredibly important for human development. Okay, so these are the known mechanisms that result in Angelman syndrome. Deletion is the majority. Mutations make up 5 to 11%. Uniparental disomy is 3 to 7%, and about 3% are imprinting defects. Now, I want to talk a little bit, let's go back. I want to talk a little bit about some of the things that have already been raised in terms of, does it matter what kind of genetics you have? And I say yes and no. Yes, it does matter. We know that, say, kids with paternal UPD may have, different ways of presenting with Angelman syndrome than children who, say, have UBE3A mutations. I don't think it's lost on anyone, if you know people in the community, that some of our individuals that have imprinting defects seem to be less affected by Angelman syndrome. And that's also true for, say, UBE3A mutations. It's not a given, right? And there are going to be families out there who have children that are having an imprinting defect or mutation that are severely affected by Angelman syndrome. So they probably hate to hear us say, oh, those kids are less affected, because they're thinking, well, my kid is still dealing with this. So it's all relative. And part of the reason for that is because there are so many other genes that make up who we are. There are a lot of things that go into making your neurons function, and it has to do with levels and amounts, and those are controlled by different things. So some of the things we do know about Angelman syndrome are the following. If you have Angelman syndrome, you will consistently have the following things. You will be predominantly nonverbal. And I say predominantly, there are plenty of individuals with Angelman syndrome who do have some words. Some have sentences. but for the most part, there are verbal issues. Movement and balance issues are consistent. Skill development is delayed. That's consistent no matter what type of AS you have. And there are abnormalities that are seen on the EEG. That's when they put the electrodes on your brain and they look at the brain waves at the surface of your skull. There are EEG abnormalities that are very specific to Angelman syndrome. That's actually how my daughter got diagnosed. When she was 11 months old, she was having seizures. We hooked her up to the EEG machine. We're having a panic attack in her room, and there's an intern in the other room. Hadn't seen my daughter at all. He saw the EEG and he said, "Hey, that looks like Angelman syndrome." And he was absolutely right. So it's very distinctive. You can actually diagnose someone by looking at their EEG. Frequently, individuals with Angelman syndrome will have seizures. And that's again because the neurons aren't functioning optimally. So the ones that are supposed to keep all the activity tamped down, they don't work very well. And the ones that are sending the activity through the brain are a little too active. So seizures are common. These are all associated with Angelman syndrome, but it's a give or take whether your kid's going to have these. Anxiety is found in quite a few kids, but not all. Some kids have sleep issues, believe it or not, some don't. There are GI issues, feeding issues, drooling, mouthing behaviors, aggression or disruptive behaviors, myoclonus and tremoring. Not all kids have all of those conditions. So we think of them as consistent conditions, frequent, associated. So this has come up a little bit, and I'll say this again. In general, there's decreasing severity as you go from the different classes through mutation, UPD, imprinting defect, and then mosaic forms. Now you're probably saying, well, you didn't talk about mosaic forms. I didn't. So mosaic forms mean that not all of the cells in the body are missing UBE3A. Some are, some aren't. There's a lot of really interesting genetics that goes into how that might happen in an individual. But individuals where some cells have UBE3A and some don't tend to be the least affected. And that probably makes sense because some of their neurons are firing perfectly and some are not. And it depends on how many neurons are missing UBE3A that probably correlates with how severe Angelman syndrome is in those individuals. But again, I want to be very clear, this is a generality. It's not always true. There are plenty of individuals who have UPD that have seizures and motor issues and are just as affected as some kids with class 1 deletions. I have met some individuals with class 1 deletions that would blow your mind. So it's not always a given that the genotype is going to tell you what your child or your loved one is going to be capable of. All right, so you might be saying, well, then what's causing the symptoms? So we know that Angelman syndrome is caused by the loss of UBE3A. And we know that the unique symptoms of AS, that impaired verbal communication, the motor issues, the seizures, that generally pleasant demeanor, that's all from the loss of UBE3A. But keep in mind, there's 20,000 other genes in your cells that contribute to who we are. I have friends whose children, one of my friends has a child who has perfect pitch. That's not because of Angelman syndrome, it's because her parents are musicians and she refuses to be in a room if there's an instrument being played that's not in tune. Our kids are amazing, and they're amazing not because of the gene they've lost, but because of all the other genes that are in there and all the amazing things they manage to do with this incredible difficulty that's been handed to them. And they don't know the difference, right? They don't know how difficult things are necessarily. I think they do figure it out. But for the most part, this is what they were handed, and they play their cards beautifully. Our job as parents is to make sure that they have the best opportunity to do that. All right, so now we're into this is completely and utterly Becky's opinion, and you can take it for what it's worth. But if I think back to when Sophie was first diagnosed, and as I said, she was diagnosed when she was 11 months old, this is what I wish I had thought about or had taken more to heart, because people did tell me this, I just didn't listen. So what are the priorities that you should be thinking about for your individual with Angelman syndrome, no matter what their age? Your number one priority is health. Do they have seizures or not? Are their seizures well controlled? Are they having GI issues? And what I mean by that is, are they experiencing reflux? Are they experiencing constipation? Are there any foods that they don't respond well to? Do they have any food sensitivities? That's really your number one priority. And the reason for that is, if you're seizing, you can imagine that when you're seizing, your brain, it's like trying to listen to the world like a radio, but the radio's out of tune. So as the person having the seizures, you're getting little bits and pieces of information, but you're not getting the whole picture because your brain is constantly fighting you. Reflux is painful. I don't know how many of you have ever had heartburn, but it is not pleasant. And a lot of people don't have heartburn for the first time until they're pregnant, and then they realize this is a very unpleasant experience. If your child is experiencing reflux or constipation, that's an incredible amount of pain, and that pain translates into things like reducing seizure threshold. So you're more likely to seize if you're constipated. I don't know if Dr. Tibert touched on that in his webinar, but we see him for seizures. And when we call, his physician's assistant will always say, is she constipated? Is she refluxing? Are her teeth coming in? It's important to make sure that the health is where it needs to be, because that's going to be the lead for everything else that happens. Excuse me. All right, the second thing to prioritize is communication. And I don't mean pushing a button on an iPad. What I mean is our kids communicate all the time. Our job is to help figure out how to make that communication useful to them. If you are lucky enough that your kid has a head shake or a head nod, make that powerful. If they say no to something, say, oh, you said no. Give them the ability to tell other people to get out of their face, right? This is, if you think about it, for me, it's really important for me to be able to tell people to go away. We've made that very important for my daughter. She can say to people, I don't want you to be in my space right now. Communication is anything that your child uses to get their point across. When our kids are babies, it's crying, right? As they start to grow up, they have words. Kids with Angelman syndrome don't have words. So we have to give them other opportunities. With our ASF series, we've got communication series. We've got people that can help you in terms of thinking about things like communication devices, but I'm really talking about everything. If your kid can sign, teach them signs. If your kid is really good at going across the room to get what they want, that's enough communication. They told you what they wanted. So focus on making their ability to tell you what they need or what they want or how they feel something that you prioritize. And the last thing are behaviors. So you want to be able to experience the world with your child, and you want your child to be able to experience the world. And so in this case, you may have to get some help if there are behaviors that are incredibly disruptive to both your individual's life and your life. This includes sleep. And I know, especially when our kids are young, they don't sleep. And we know this. And I remember telling my pediatrician that Sophie had gone for 48 hours without sleeping when she was about a month old. And he told me that wasn't possible, that I probably had just slept through her sleeping. And I'm like, trust me, I didn't sleep through her sleeping. She wasn't sleeping. It's important to try to help our kids sleep. And there are neurologists and psychologists that specialize in sleep. If you have the ability to find one, do it. Because our kids can learn to sleep, it just takes them longer, and they may need assistance. All right, that's my thoughts about what you should focus on for your kids. What about for you? It is as important, if not more important, for you to focus on yourself as a caregiver. Caregiving is hard. It is hard, and people say we're super people, and that's all great, but the truth of the matter is you've got to take care of yourself. You can't take care of anyone else if you're not taken care of. So your health is #1 priority. That's mental, physical. Do anything you can do to reduce stress and guilt in your life, because having a special needs child is basically given a great big basket of guilt that you carry around all the time. And it's exhausting. And it's really hard on us. So it's important that you do whatever you can to make sure that your health is taken care of. Try as best you can to get some sleep. And I know that you're probably out there laughing and I can't see you. What my husband and I did early on is we just traded off. One person was up, one person slept, and we just traded off. And we did that for years until we could get her to sleep. Whatever you can do to get some sleep, try to do it. And I know in some cases that's going to be really, really difficult, but sleep is important for you as well as your child. And the other thing is find community. We have a great community in the Angelman syndrome community. We're really lucky in this particular regard. The people in this community are amazing. They will reach out to you. You will make some lifelong friends. There are people that are willing to drop everything and help you, or call doctors for you, or find people to help you, or even help you reboot your iPad when you don't know how to do it. This is a great community. Make use of it. Join the Facebook groups, join the parent communities that we have, join the various different parent organizations here and across the world. Don't do this alone. No one needs to do this alone, and we're here for you. All right, one set of stress I would like to take away from everybody right now is the idea that you did something wrong that caused Angelman syndrome. What I mean by that is this. So if we go back to our little diagram here, here's mom and dad, here's mom's egg, here's dad's sperm that make the baby. So if you think about it, as moms, we've got this little guilt too, that it's our DNA that's causing this problem, right? It's the DNA that we gave our child or didn't give our child in the case of UPD. I want you to realize that this egg that came from you that makes your child actually developed and came into existence when you were still in utero in your mother. All right, so let's be very clear about this. When you were in your mom's belly, when you were in your child's grandmother's belly, you made those eggs. So before you even became in existence in this world, your child was sitting there ready to be born. I don't mean that in any sort of spiritual way. What I just mean is that you didn't do anything to cause this. There was no possible way for you to do that. You didn't drink anything. You didn't walk past anything. You didn't, partake of whatever at some party in college. Nothing that you did caused your child to have Angelman syndrome. I don't know if anybody needed to hear that tonight, but if you did, let me just take that guilt right away from you. Nothing you did cause this. The other thing I want to leave you with is this idea that was brought to me very early on in the Angelman syndrome community, and it took me a while to sort of get on board with it, so it's okay if you're not there. But it's this idea that words really, really matter. How we talk about Angelman syndrome matters, not just to people out in the world, but to our kids, whose ears work just fine, by the way, but also to ourselves. How we talk about Angelman syndrome and how we think about Angelman syndrome influences how we feel about it. And this will sometimes really hit you when it's a doctor or a researcher or someone involved in industry that says something and you think, ooh, that really didn't land well. I don't like what you just said. Words matter. Try not to take it personally when someone says something that's really annoying to you, but it's okay to educate. And I just want to share this story with you before we take some questions. So I would really love it if we could get people involved in our community from biotech to parents to start using this language. Our kids are either strongly affected by Angelman syndrome or they're less affected by Angelman syndrome. My daughter, strongly affected by Angelman syndrome. I have some friends whose children are less affected by Angelman syndrome. This is such better language than saying, my kid is severe or low functioning. Oh, what a horrible thing to say. My kid is less severe, my kid is higher functioning. That might feel better, but there's a comparison there that's really, really derogatory. Angelman syndrome just is, and it either really affects our kids or it doesn't. These are words that sort of imply what our kids are capable of, and they're damaging. And the reason I know this is because I had an experience, because I'm a scientist. I went to a scientific meeting, and researchers in Angelman syndrome happened to be on the panel, and they were presenting some work that they had done in the natural history study. And I realized sitting in the room that one of the individuals they were talking about was my daughter. And the way they were talking about her in that room brought me to tears because I realized that they were saying, our research shows that individuals like this, my daughter, are less capable cognitively of functioning essentially in the world. So here's how my daughter presented when this study was presented. She started seizing around 7 months. It probably was earlier, but that's when we recognized it. At 3 to 4 years, she was able to sit up without support, but she couldn't do it for more than a few seconds. Even now, she can sit for about 30 minutes, but you have to be there with her because she has no feeling or balance. She only walks if you completely support her. She's never taken an independent step. The number of seizure drugs that she had been on, right now it's 2, but she's tried 6. And she took this test that I absolutely loathe called the Bailey's, and she scored the lowest possible score it was to score on this test without being, say, a houseplant. So I'm sitting there listening to them talk about this, and I'm realizing that I'm internalizing this information, and I'm thinking, wow, my daughter is like the most impacted kid with Angelman syndrome that has ever existed. I'm going to show you a video, and it's going to be really sort of shocking, but let me just show you what this incredibly affected kid looks like. All right, here she is. My gosh, you're going fast. So this is my severe low functioning Angelman child. And what she's doing is she's riding her bike, which is her favorite thing to do in the whole world. And what you can tell because she's laughing. What you can see here is a guy is walking in with her toddler. The thing that she enjoys more than riding her bike is running over toddlers with her bike because they make really great noises when you run them over. So now watch her. What she's doing right now is she's planning how to get away from me and go hit that toddler. And you can see it because she slows down when she sees the toddler and she's circling around. This video is going to end because she's going to go after the toddler, which I kind of knew she was going to do, and I had to go get her. That's my low functioning child that was presented at this webinar as having no cognitive function. Words matter. She's not that kid. And it's funny because people who know her or have seen her on Facebook or things talk to me all the time and they say, well, it's not fair because Sophie's so high functioning. Like she's very strongly affected by Angelman syndrome, very strongly affected, but she's amazing. And the way that we talk about our kids changes how we feel about them. It changes how other people feel about them. And it impacts them because they hear us. So that's my general soapbox that I'm going to jump off of now. But I want to stop here and see if people have any quick questions and I can answer them. And if we have time, then we can go on and talk about clinical trials.
Yeah, so we'll answer a couple, and I know that your clinical trial section's pretty short, but I think it's important to get to. So
Okay.
just really quickly, there's a question about when we talk about genetic testing, can someone go back and obtain the size of deletion if they would like to?
Absolutely. Yeah, you could go back and ask to have the test run again, specifically for the size of deletion. If you need a medical justification for that, you just ignore everything I said in the seminar and you say, it's important for me to know the size of the deletion because it could change what kinds of care I give my kid. All right.
Perfect.
That's what you say.
Yes.
In your head, you say, whatever it takes to get my test done.
Exactly.
But don't believe it. Okay.
And then which transmissions are hereditary?
Aha.
So in which do you need to be concerned about that the individual with AS siblings may have a child with AS?
That the individual's siblings may have a child with AS?
Yeah, so it says, in which do you need to be concerned that individuals with AS siblings may have a child with AS? So I'm guessing that if, like, for instance, Burdine, who is Jackson's brother, would they have a child? Could they have a child with AS?
Burdine's chance of having a child with AS is the exact same as anyone else walking the planet. It's about 1 in 20,000. So if he has 20,000 children, one of them probably will have AS.
Perfect.
So the reason for this is the deletions happen in the eggs. And if you had one of these deletions, you would either have Angelman syndrome or Prader-Willi syndrome. Now, we do have individuals in our community whose parents had Prader-Willi syndrome and their children had Angelman syndrome. And I'm happy to walk you through that if that seems really crazy. But most individuals with deletions don't have children. Deletions are not hereditary for the most part, and I'll come back to that in a second. Mutations are the ones that you have to worry about, and imprinting center defects are the ones you have to worry about for potential transmission for additional children that you have. Your children who do not have Angelman syndrome have no additional higher chance of having another kid with Angelman syndrome than anybody else on the planet. So in the case of a mutation, you want to get tested so that you as mom want to get tested to see if you have that same mutation in your DNA. Because if you received that mutation from your dad, where the gene wasn't used anyway, it wouldn't have any consequence in you, but you could pass it on to your kids half the time. So you want to be tested and you want to see if you have the mutation. A lot of our kids with mutations, they're called de novo mutations, which means it was in the kid's DNA, but it's not in mom's DNA. So then your chance of having another kid with UBE3A mutation is very slim. Deletion, the only time that this might be something you could pass on to another child is if you have something called a translocation. So if you have a child with a deletion, you should still ask to have your DNA tested to see if you have a translocation yourself. That's way too complicated to get into on this seminar, but most people will not have this translocation. and your chance of having another child with Angelman syndrome is 1 in 20,000. So you would have to have another 19,000 kids.
Yeah, and Win Han just brought up too, it's always really good just to talk to your clinical geneticist or your genetic counselor
Absolutely.
about all of this, just because they would be a great resource for you to be able to talk through if more testing needs to be done, to be able to do that.
Absolutely. And they will help you get the testing you need. So your clinical geneticist, this is absolutely what they should do. If you go to any of the ASF clinics, there are clinical geneticists like Dr. Tan who could help you with this because they're really familiar with Angelman syndrome and all the testing that's out there.
Yeah, and kind of along that, Becky, one of the questions that came up was we have a lot of people who have a clinical diagnosis but have never gotten genetic testing. So one of the questions came up was, do I see a geneticist for testing, and can I do that in Angelman Clinic?
Yes, you can. I would absolutely see someone for testing, and the reason for that is that we have so many clinical trials coming on the horizon, and to participate, you need a diagnosis. You need a clinical diagnosis. A genetic diagnosis. And so there is a rationale that should allow for insurance to pay for you to get tested because there's a potential that there could be a treatment in the future that you need to know if you are eligible for. It's also important, I think, at this point, because if your child has something that looks like Angelman syndrome but isn't, such as Pitt-Hopkins or one of the GRIN 2B types of syndromes that are out there, you're missing that community too. Right? You can always be part of our community because you're here, you're clinically diagnosed, everything that we're dealing with is very similar, but you don't want to miss out on being in those communities because they may have trials that start at some point and you want access to those. So I think it's critical to go ahead and get your testing done. And that is true even if your child is an adult, because some of these trials have been, there for adults. And I would absolutely recommend doing that.
So we have a plethora of people that are telling you, thank you, you're amazing, I'm learning so much from this. So I just wanted to point that out. But if you could, we're running low on time. So if you could I think it's important to move forward a little bit with what you were going to talk about. Do you think you could maybe breeze through it in the next few minutes?
How many minutes do we have?
Well, it's up to you. People can stay on, they can leave. Let's just move forward.
All right, so this is just some information I want you to have because we are in this brand new area where we have clinical trials coming on the horizon. We have clinical trials happening right now. When I first got diagnosed with Sophie, I had a conversation with Dr. Art Baudet, who told me this was coming in 10 years. I thought he was crazy, but he was absolutely right to the year. This has sort of been my dream since Sophie was a little kid, is that I wanted industry to really look at Angelman syndrome and see that there was this huge potential for therapeutics and to get on board. And now they all are, and we are so blessed to have all of these different companies doing this work. So some things I want you to know is, first of all, we already have therapeutics for Angelman syndrome. And you might be thinking, what? That's not true. It is true. We have a lot of therapeutics for Angelman syndrome that we know work, that have been researched and work well. And they include anti-epileptic drugs and diet for epilepsy, like the low glycemic index treatment. There are behavioral therapies and medicines that help with sleep. There are reflux medications. There are behavioral therapies, anxiety medications that help with behavior. Physical and occupational therapy, probably all of our kids have done this at some point, if not still. And for communication, we have speech therapy, education, iPads, all kinds of things. These are therapeutics. These are all things that help our kids progress and have a better quality of life right now with Angelman syndrome. So when we're talking about more therapeutics, we're talking about things that are a little more targeted. And in this case, what we mean is putting UBE3A that's missing back in those neurons. Can we do that? And the two general strategies that are out there right now, one is to replace that UBE3A that's in neurons. And we can either do that by using viruses to put the UBE3A gene back in the neuron so that they can now make protein, or we could try to turn on that paternal copy of UBE3A that's just sitting there uselessly hanging out. We need it to turn back on and function. And a lot of the industry updates that you can see on the video at the website, we're talking about doing this, and they do it in various different ways with things called antisense oligos, ASOs. LNAs, there's CRISPR approaches, there's lots of approaches that are out there that are all very exciting from lots of different companies that might get this paternal UBE3A to, quit loafing around and maybe turn on and help our kids a little bit. The other approach is to try to correct the function of those neurons in the absence of UBE3A. So this is trying to find drugs or compounds that can improve the way the neurons function. And the truth is, we may need both of these things in combination. We may need to put UBE3A back as best we can, and then use some of these drugs to help neurons function better, even though they may not have the full amount of UBE3A they need, but maybe they get a little bit, and we make the neurons function better, and we get kids that can learn better and faster and can correct some movement. There's all kinds of things that could happen here. So as a scientist, I'm super excited about all of this. And I really do think you should go watch those industry update videos, especially after hearing Genetics 101, because I think some of that will make more sense. Is it even reasonable to think that we could do this, right? So how do we know that even putting UBE3A back would do anything that's important? Well, I'm going to give you 3 examples. The first was this paper published in 2001. And in this case, what happened is they took a mouse with Angelman syndrome and they combined it with a modification in a different protein entirely called CAMK2. Doesn't matter about what it does, it's just involved in learning and memory. But the idea here was that we're missing UBE3A and we changed the way CAMK2 functioned. And when those mice were born, they did not have Angelman syndrome. The loss of UBE3A didn't really impact the mice. So what this told us was that if we could improve neuronal function, we could deal with not having UBE3A. So that's the second strategy, right? How do we improve neuronal function in the absence of UBE3A? So that worked in mice. The other thing here is that this protein they modified, CAMK2, is only expressed in the brain after birth. So basically, after all the development happened, this change was made and we still saw a benefit. So that suggests that there aren't defects in how the brain develops in Angelman syndrome, there are defects in how the brain functions. That's easier to deal with. All right, so in these 2 papers, it was shown that if you could restore UBE3A protein to the adult AS mouse brain, you could actually rescue phenotypes that were in those mice. So mice that had phenotypes that looked like Angelman syndrome, they got protein in their brains, they looked like they didn't have Angelman syndrome phenotypes anymore. Now, this study is really important because it did say that some phenotypes didn't look like they got rescued, but others did. And so that might mean that we might be able to put UBE3A back and we're still going to need some intervention, right? I don't think we're going to put UBE3A back in the brain and the kids are going to stand up and start singing. I think they're still going to have some therapy and things to learn how to talk if this works. So there's a lot here, but the most important thing was that we could rescue things like neuronal plasticity, which is the ability of neurons to learn at any age in the mice. All right, so these are just 3 out of many, many, many papers, and I just cherry picked these 3, but there are others. So what does this mean? Well, here's the bottom line. If you're a mouse with Angelman syndrome, we have very good news for you because we can fix it. Mice aren't humans, but there is absolutely no reason to believe that targeted therapeutics are not possible, and we're not going to know until we know. And to know, we're going to need clinical trials because we know what we can do in mice and rats, and there are some studies that are going on in primates, but we're not going to know what this can do for humans until we try it in humans. All right, so what do I want you to know? Clinical trials, phases of testing. So there are first the studies that are in the lab and studies in animal models. This is all the discovery phase, and that's where a lot of our science was for most of my daughter's life. But now we're moving into clinical trials. And you can learn a lot more about this at various websites that I'll show you in a minute. But I just want to point out that there are phases. You start first to see how does a person do with a potential therapeutic intervention. Then you do a small number to see what the safety is of that intervention and try to figure out what dose you need of the intervention. Then you move into the phase 2 trials, which is more about trying to assess how a patient is responding to the intervention and if it's still safe. And then finally, you move to big stage 3 trials where you have lots of individuals who use the therapeutic intervention and you try to measure how effective it is compared to other things that are out there, or if there's nothing out there, how effective it is, period. You're going to hear a lot about this called outcome measures. Outcome measures are tests that accurately determine how you're functioning before any treatment's provided. So how are you doing with your motor skills? How are you doing with your communication? How are you doing with seizures? How are you doing with sleep? Anything that basically says, here's where I am at baseline, and then I'm going to use that to test during treatment or after treatment to see if any of that changed. So that sounds relatively simple, but it's really not. So how do you know that a therapeutic actually helped someone if you can't measure it? And what exactly are we going to measure? And one thing that's been frustrating for me trying to learn about clinical trials in some of the things I've been doing is that the FDA wants outcome measures that they know will work. But if we don't have any clinical trials that have been successful, we don't know which ones are going to work, right? So you need something to be successful to know if the outcome measure worked, but you need the outcome measure to know if the thing was successful. So we're still in a little bit of a gray zone here, but we're working on outcome measures. People are trying to develop new ones. We're trying to assess the ones we actually have and see if they're going to work. But you're going to hear a lot about what are the outcome measures that are used. And these are essentially the tests that are used in trials in order to determine whether or not the trial worked. In a very basic trial design, you sign up for the trial, you pass a screener, and then you're randomized. And this randomization is done by a group of people that are not involved in the trial in any other way. So that the people running the trial, the physicians conducting the trial, nobody knows whether your child or loved one is in the treatment group or the control group. This is critical, and I'm going to tell you why it's critical in a minute. But in a very basic placebo-controlled trial, this is how it's going to go. You're going to be in treatment or not, and you're going to take those outcome measures, both at baseline and during the trial. All right, so why do we need a placebo? So I call this, I know myself and I know my child. Absolutely you do. There's absolutely no way on earth that you don't know your kid better than anybody else. The reason we need placebo-controlled trials is this incredibly freaky thing that you have no control over. And it's your brain and this freaking weird thing that happens. We know from many, many decades of work that there is absolutely no way to prevent your brain from making connections between patterns that you see. And if you know that your kid is on a therapeutic drug, you will make connections that make that therapeutic drug positive. And you can't help it. This is not about what you know or what, knowing your kid or anything. This is about something that's encoded in us genetically that we can't control. And here's an example of this. There was a trial that was done for Parkinson's syndrome. And in Parkinson's syndrome, you're missing levodopa in the brain. In this particular trial, they had to drill holes in the skull to do the intervention. And so the placebo control group had holes drilled in their skull, and they had surgery sounds played like they were actually having the surgery. The only thing they had done was the hole drilling. The placebo controlled group, some of those individuals got up and walked across the room after surgery when they had been unable to do it before. They had not had surgery. They had been tricked into thinking they have surgery. If that can happen to them, there's no way any of us, physicians, researchers, parents, we cannot possibly be objective. We just can't do it. Our brains won't let us. So we need placebo-controlled trials so that we know that anything we see is real. And that's why we need it. This is funny, but not funny. A lot of drug companies will tell you that we should just be studying placebos because the placebos always tend to work really, really well. The flip side of the placebo is something called the nocebo. This is where there's a harmless thing that you are convinced causes harm because you believe it's harmful. It's the exact same reverse. It happens to us all the time. If you eat something and it makes you sick, every time you see that thing, you'll feel like you're sick. If you are convinced that when your child eats X, Y happens, that will always be the case. It's something you can't control. So we know in clinical trials that there are plenty of people who report adverse side effects, and they're convinced that these are horrible adverse side effects, and they're on placebo. So it's just something you can't control, but that's why we have to have it, and that's why we have to participate, even though we might get placebo. So what do we do? Well, in God we trust, we have to bring data. We have to have measurable things. And some of the difficulties are this: we're a small community. And so if every clinical trial out there needs at least 100 participants so that they can do a placebo-controlled trial and get data, you start to see the problem, especially when you start to have inclusion and exclusion criteria. How do we get 100 people with Angelman syndrome of this particular genotype with these symptoms who can participate in this trial and can be there for all the visits? It gets tough. right? And because we have a small community and we have a small number of people that can participate in trials, the change we have to see between those on placebo and those on therapeutic has to be big enough to measure. And to put this into context, some of the really popular antidepressants that are out there on the market that have been proven to work, they were only proven to work after they were tried in thousands of people because the effect was so small. If they'd been tried in 100 people, they would never have made it to the market. So this is going to be a problem. And it's not anything we can do, anything about it. All we can really do is participate. This is the last difficulty I want to tell you about, and that's the fact that our kids, even though they're consistent and frequent and associated things, every one of our individuals is different. And you all know this. If you've met any other kid with Angelman syndrome, they're all different, right? You know, my Sophie, like I said, she's never taken an independent step. She's not anxious. She's never been anxious about anything in her life. I think she's the most laid back thing on the planet. There are other kids with deletions that are her age that walked right on time and everything scares them to death, right? How do you do a clinical trial when learning to walk would be a miracle for my kid and no change for this one? This is tough. All right, so what can you do? As a parent, as a loved one, as a sibling, get informed. The FDA has a great website that's just very general. What do you need to know, right? And it's the FDA that's telling you this. So nobody's got any, you know, this is just straight talk from the FDA. Here's what you need to know. This is an amazing website that's been put together by a lot of the parent groups and the industry groups here for us, Angelman clinical trials. It has the trials that are out there. It has a lot of the information that you'll also find on the FDA website. These are the two places where you go for information. You have to participate. Like, none of these trials are going to work if we don't have people that are involved. But there are issues with participating. There are, you know, risks that we're taking. There are all kinds of things you have to think about. So I want to make it very clear that you can do all kinds of things to participate without being in the trials, but you also need to be in the trials. Save your medical records. We have plenty of them, right? So now they can be useful. Visit the AS clinics. Get involved in the natural history study. You might say, well, why is the natural history study helpful? When we have so few people, the FDA will let us say, here's what happens to people in the natural course of Angelman syndrome, and we've got all those records from the natural history study. That's our placebo group. What happens to these individuals that are doing therapeutics? There are ways that the natural history study can help leverage the numbers of individuals we have that can participate in trials. So it's critical to be involved in that. Participate in the surveys, all of the surveys that come around, fill them out. And then contribute to those ongoing studies and trials. It's critical that we do this. Always consult your medical providers about any trial you want to participate in and read all the paperwork. That's just basic common sense. Do those things. Don't participate in anything until you have a full understanding of what you're agreeing to, what's happening, what's expected of you, and what your options are. You can find other information at clinicaltrials.gov, and this will list the trials that are involved with Angelman syndrome. Sign up for the newsletters from your parent support organizations. Follow all those companies that are in that video on industry reports. Follow them on social media because they'll tell you what's happening. Get involved in the Facebook community so that you get the news, so you know what studies are out there and you know what surveys are out there. And this is the last thing I'm going to leave you with. In the Fragile X community, there was a compound that looked very promising for clinical trials. And the inclusion criteria to be in this trial is your child had to score a certain way on the inclusion survey that came out to show that they had a very high level of anxiety and irritability. So the community got on Facebook and they helped everybody figure out how to game that survey so their kids could get into the clinical trial. But a lot of those kids didn't meet the criteria for the clinical trial. And so when the trial data was unblinded and they looked to see if there was actually a result, it got washed out by all the individuals who shouldn't have been in that trial to begin with. And even though this drug did some amazing things for the individuals who were having a lot of anxiety and aggression, they couldn't move that drug forward because their clinical trial essentially failed. And it failed because everybody wanted to get in and beat the inclusion and exclusion criteria. Don't do this, right? Some of us won't meet the inclusion criteria. My daughter's unlikely to meet the inclusion criteria for a lot of these trials because she can't walk. That's really frustrating for me, but we have to do these trials correctly. So if the trials say don't share things on Facebook, don't share things on Facebook. If the trials say this is the inclusion criteria and your child ends up not meeting it, you know, stamp your feet and be mad for a minute, but think about the fact that we have to do this the right way if we want those drugs to ever be available to all of our kids who can't get into these trials. So we can do this as a community. Don't get in our own way. All right, that's all I had to share with you guys tonight. I appreciate your attention.
Yes.
Go ahead.
Yeah, so thank you so much. We do have a few questions, but we are way over time. So Becky, I'm hoping that I can convince you to complete those questions for us, and we will get them out to the community along with your presentation. Amazing information, Becky. I learned so much tonight. I can listen to you for hours. Thank you for putting all of this into terms that we can understand, because it's really, really hard as a parent coming into this space to hear all these things being thrown at you, and you're like, I don't even know what that means, but you're just kind of nodding your head because you're still trying to deal with the diagnosis. And so thank you for taking the time to share that with us. And you know, Becky is I would say Becky is an individual that if you ever have a question or want to hear more or want to understand more, she is so willing and has been such a vital part in this community since day one. I'm sure she'd be more than willing to share, and we will definitely hear from her in future presentations as well. So Becky, thank you so much for being with us tonight. Thank you for having me.
Thank you for staying late. I appreciate it.
No problem. So we will, just so you guys know, we're going to take a break for a couple nights. So you have your Saturday and Sunday off, but please come back on Monday. Monday, we're going to kick off with Monday evening being a session all about puberty in Angelman syndrome. This has been a hot topic, right, Becky, in the space of Angelman syndrome, of what do we do when our kids are starting to hit puberty. So we're going to have clinicians from the Angelman syndrome clinics to come in and talk to us about this, and it'll be great resources. And then stay tuned for the rest of the Virtualpalooza for the rest of the week. Until then, have an amazing weekend with your families. We love you, and we thank you so much for being with us tonight. Thank you, Dr. Becky Burdine, and guys, have a great weekend. Thank you, guys.